Can everyone hear me? Thanks. Hey.
I was supposed to talk back live.
Oh.
Thank you. Appreciate it.
Afternoon, sports fans. We will finish up the Krebs cycle today and then head into the very interesting subject of oxidative phosphorylation. Let's begin, as we should always, with a word of prayer. Lord, we thank you for the bright shining sun outside, even though it is cold. We thank you that the sun for us represents warmth and life. We pray now that you would be our warmth, our life amid these less than ideal circumstances. We pray now that you would watch over us, help us to learn, help us to not complain, but to reach forward, work hard, be thankful for what we have. In Jesus' name, amen. So I did not go through this last time, but this illustrates in cartoon what is actually happening here. So the triacylglycerols, remember, those are fats. They are hydrolyzed to fatty acids and glycerol. Glycerol is not an issue. That can go straight to the cytosol and play a role as a metabolite. The fatty acids are then transported into the peroxisome/glyoxysome. You have beta oxidation, which converts the fatty acids into acetyl coenzyme A. Acetyl coenzyme A enters the glyoxylate cycle, of which the first 3 steps are exactly the same as the Krebs cycle. You condense acetyl coenzyme A with oxaloacetate with the help of citrate synthase. Citrate is then converted into isocitrate by a conatase. What? What was that? Somebody said something.
I'm going to go with an accident.
Oh, it was an accident. Okay, never mind. All right, and then the isocitrate is subjected to isocitrate lyase. That will give us succinate. Another acetyl coenzyme A, again, from beta oxidation, is fed into this. It condenses a glyoxylate, malate, with the help of malate synthase. Malate is then oxidized by malate dehydrogenase, and the whole thing repeats again. Succinate is shipped to the mitochondrion. There, it will enter the Krebs cycle, undergo hydration by fumarase to fumarate. Fumarate will then it undergoes oxidation via succinate dehydrogenase to fumarate. Fumarate then is hydrated by fumarase, which makes malate. Malate then is oxidized by malate dehydrogenase, going to oxaloacetate, and the whole thing starts all over again. Malate can be withdrawn from the mitochondrion into the cytosol. Malate will then be oxidized by a cytosolic isozyme of malate dehydrogenase to oxaloacetate. Oxaloacetate is then the beginning substrate for gluconeogenesis. You'll convert it to phosphinylpyruvate via either phosphinylpyruvate carboxylase or phosphinylpyruvate carboxykinase. And then the process goes in reverse and you make sugar. If that sugar is going to be exported, you'll convert that sugar into sucrose. If that sugar stays put, you may not convert it into sucrose, but sucrose is the way you ship things around cells in the plant, and that's the major component of sap is sucrose. So as I said, the galactose pathway is found in bacteria, it's found in green plants, it's not found in vertebrates. Vertebrates lack malate synthase, they lack isocitrate lyase. The common enzymes are isozymes of each other, cytosmic and mitochondrial versions of malate dehydrogenase, and so on and so forth. And vertebrates lack the glyoxylate cycle. The glyoxylate cycle partitions certain reactions to certain organelles. You then have coordination between those organelles for the co-regulation of the glyoxylate and citric acid cycles. And in germinating seeds, this is precisely what you have. Fatty acids are oxidized to acetyl-coenzyme A. Acetyl-coenzyme A is used to make sugar. We can't do that in our bodies. Plants can because they have the glyoxylate pathway. We reviewed this before. Again, succinate goes from the galactosome to the mitochondrion. It's converted to malate. Malate goes to the cytosol where it's used for gluconeogenesis. Very good. All right. So oxaloacetate is then moved from the mitochondria in the form of aspartate. Aspartate. And someone's having trouble getting into the Zoom session.
I think that might be Greg. He's texting me right now. He said he
Okay, he texted me and I couldn't tell who the text was from. Sometimes it helps if you log off and log back in. I don't know why it does that, but sometimes that helps.
I had to sign in. Like I had to sign into an account. It won't let you use it without an account.
Does Greg have an account?
I don't know. He should be able to like log in with Google though.
Yeah, that too. So if someone could please text him and let him know, give him some help, maybe he could.
Yeah, I'm texting him right now. So.
Okay, terrific. He actually showed up here. He's forgotten that we were online. So, oh well. Okay, and the aspartate just gets reconverted into oxaloacetate. It's just a deamination reaction. It's really quite straightforward. All right, so the coordinate regulation of these two pathways depends upon isocitrate. Isocitrate is a branch point. You either take isocitrate, you subject it to isocitrate dehydrogenase, in which case you go through the Krebs cycle, or you subject it to isocitrate lyase, in which case you go through the glyoxylate cycle. So isocitrate is the branch point. And isocitrate dehydrogenase is regulated by covalent modification via a protein kinase. When you phosphorylate it, you inactivate it. Phosphorylated isocitrate dehydrogenase is inactive. That now directs the isocitrate to the glyoxylate pathway, where it's used to make glucose rather than making NADH for ATP synthesis. Dephosphorylation of isocitrate dehydrogenase reactivates it. And now we'll shunt your isocitrate through the Krebs cycle. It'll make NADH, it'll make ATP, and so on and so forth. Turns out the isocitrate kinase, right? Isocitrate dehydrogenase kinase and isocitrate dehydrogenase phosphatase reside in a single polypeptide. It's a one protein sort of deal. And it is regulated by various intermediates. So both the citric acid cycle and glycolysis intermediates stimulate the phosphatase activity. If you stimulate the phosphatase activity, you also inhibit the kinase activity. And that is going to activate isocitrate dehydrogenase that activates the citric acid cycle. So these intermediates of the citric cycle and glycolysis not only activate the phosphatase activity, they also inhibit the kinase activity. Sorry, they're also inhibitors of isocitrate lyase, which is the first enzyme in the glyoxylate cycle. So citric acid and glycolytic intermediates are going to promote TCA cycle and ATP synthesis. The logic behind this is that if the energy producing metabolism is working quickly, then the buildup of glycolytic and TCA cycle intermediates will decrease. If that's not the case, then these intermediates will build up. They'll be able then to regulate the TCA cycle such that you want more of it, so you have more ATP. Isocitrate lyase inhibition is relieved and the protein kinase is no longer inhibited if the energy metabolism is moving along fast enough. In that case, you don't have sufficient quantity or concentration, shall we say, of Krebs cycle or glycolytic intermediates. In that case, the isocitrate lyase inhibition is not robust, and the protein kinase is no longer inhibited. And these conditions will inhibit isocitrate dehydrogenase, and isocitrate then gets shunted to the glyoxylate cycle for hexose synthesis. Essentially, if the energy yielding processes are working fast enough, that means you're making lots of ATP. If you're making lots of ATP, then you can afford to use it, because making glucose is very energetically expensive. If you have enough energy, then you're going to use it to make glucose. If you don't have enough energy, then you're not going to run the glyoxylate cycle. You're going to run the TCA cycle until you get enough energy, then you'll be able to use it. That is the logic, if you will, of the regulation of the glyoxylate and citric acid cycle in the plant. Okay. And now, Gregory, glad you could join us.
Yes.
Okay, now for something Completely different. No. Hey, Manda, I saw that. There we go. And are the slides moving? Does the slides move?
Yes. Yep, they're
Good.
moving.
They're moving, good, okay. All right, oxidative phosphorylation. So as we said, these oxidative processes, glycolysis, citric acid cycle, these cycles are convergent, they're oxidative, they produce lots of reduced carriers and lots of reduced carbon in the form of carbon dioxide. So you make 6 ATP, 4 from glycolysis, 2 from the Krebs cycle, 10 NADH, and 2 reduced flavings per oxidized glucose. If you want to oxidize more glucose, you must reoxidize those carriers. That's not a trivial thing. There's a lot of energy in those high energy electrons that are carried by reduced flavings, NAD, something like that. If you ferment, it's easy. You just take your carbon skeleton, dump those electrons there. And then you essentially excrete a reduced carbon compound, a reduced acid, a reduced alcohol, something like that. So the compounds that are excreted in the case of fermenters are always more reduced than glucose and less heavily oxidized than carbon dioxide. Electron transport chains are another way around the problem of re-oxidizing the carriers. And the electron transport chain will kill 2 metabolic birds with 1 beautiful cellular architectural stone. Namely, it will not only reoxidize the carriers, it will also provide the opportunity to make large quantities of ATP. This is the process we call oxidative phosphorylation, otherwise known as cellular respiration. You're going to do this by creating a proton gradient on one side of a membrane, a proton impermeable membrane. And that gradient gives you a disparity in concentration, charge, and pH. That gives you the capacity to do work, to do enormous amounts of work. That work will be used to drive an F-type ATPase. So that's where we're going. This is oxidative phosphorylation. All right, in 1948, Eugene Kennedy, a very famous biochemist, and his student at the time, Albert Leninger, yes, Leninger of our textbook, he showed that mitochondria are in fact the site where oxidative phosphorylation occurs in eukaryotes. Leninger did not work out the mechanism by which it occurs because every time he took mitochondria and broke open, ATP synthesis ceased. He kept looking for some sort of high energy intermediate. He never found it. And it never quite dawned on him that the membranes, in fact, were what was necessary. And it took Peter Mitchell essentially to take where Leninger had left off to figure out how mitochondria and bacteria and chloroplasts all make ATP using proton gradients. So this is a nice little cartoon of a mitochondria. So there is, first of all, an outer membrane. That outer membrane is relatively permeable. The outer membrane can let small molecules, small ions, maybe peptides, things like that through it. It can't let proteins through it, but it's selectively permeable. The inner mitochondrial membrane is a bird of a different feather. This membrane is quite impermeable to most ions, most small molecules, and the inner membrane, inner mitochondrial membrane, contains bucket loads of protein. Lots of protein. You can see the things this membrane contains. It contains the elements of the electron transport chain. It contains the ATP-ATP translocase. It contains the ATP synthase, that we could say the F-type ATP synthase. It contains lots of other membrane transporters. And then the matrix contains your citric acid cycle enzymes, pyruvate dehydrogenase complex, enzymes of fatty acid oxidation, beta oxidation, which, by the way, those occur in the mitochondrial matrix in animal cells, not in plant cells. In plant cells, fatty acid oxidation occurs in peroxisomes/glyoxysomes. It also contains enzymes for amino acid oxidation. It also contains its own genetic system. It has a circular chromosome that's somewhat bacterial looking in its organization. It also has ribosomes that are very bacterial in their architecture. It transcribes that DNA. It replicates that DNA. It also contains a host of other enzymes, metabolites, soluble stuff, and so on and so forth. So if there is a problem with mitochondrial DNA, you have a mutation, let's say, in mitochondrial DNA, you will usually suffer from severe muscle and brain degenerative diseases. These will usually be inherited from your mother, and you typically won't live very long except under certain unusual circumstances. Mutation in one such gene, for example, the BCST1L gene, which is required for assembly of complex 3, causes a so-called Gracile syndrome. Gracile syndrome. So that's G-R-A-C-I-L-E. G stands for growth retardation. R stands for, well, GR is growth retardation. A is amino aciduria. C is cholestasis. I is iron overload. L is lactic acidosis. And E is early death. Boy, that doesn't sound like it's any fun, does it? That's just one of the many different types of syndromes caused by mutations in mitochondrial genes. Let's look at the components of the electron transport chain. So electrons will be given to the chain in the form of reduced NAD. And electrons will also be given chain in the form of reduced flavine. So there's two ways to enter the chain, either with FADH2 or NADH. And there are two ways to enter the electron transport chain. So those are the carriers that bring the electrons, high energy electrons, to the chain. NADH, a result of NADH-linked dehydrogenases. And you reduce NAD to NADH. NADPH is used typically for anabolic reactions, and ADPH tends to be produced in the cytosol on the average. There are a few exceptions to that, but that's generally true. As we said before, NAD-linked dehydrogenases transfer two electrons and a proton to the NAD to make NADH. The H is carried in the form of a hydride. The hydrogen ion goes free into solution. And NADH and NADPH are completely interchangeable inside cells due to the activity of this enzyme, nicotinamide nucleotide transhydrogenase. And it catalyzes this reaction in either direction. So an NADH is easily an NADPH, and NADPH is easily an NADH. You can swap the two, and you can swap the two easily. NADH cannot cross mitochondrial membrane, neither can NADPH. Instead, they're shuttled across the membrane by other means. Let's take a look at a particular shuttle that is used by cells to bring cytosolic NADH into the mitochondrion. So let's take a look at our makeshift blackboard here. All right. So let's say that this is the inner mitochondrial membrane. There. That's the inner mitochondrial membrane. Then let's say that this one here, let's say that's the outer mitochondrial membrane. And then out here, we have glucose, goes through glycolysis, and it makes glyceraldehyde 3-phosphate. And this is going to be subjected to glyceraldehyde 3-phosphate dehydrogenase, which is going to take NAD plus an inorganic phosphate. And it's going to produce from that reducing equivalents, and from that you're going to get (1,3-bisphosphoglycerate), and then that eventually gets converted to pyruvate. So that's colysis. We know that pathway quite well. Now, how do we go about renewing essentially this so we can use this for, so here, over here is your, let's call this the matrix, mitochondrial matrix here. This is going to get oxidized back to NAD by means of an enzyme that is called glycerol 3-phosphate dehydrogenase. Glycerol 3-phosphate dehydrogenase. So this is GPD1, otherwise known as glycerol 3-phosphate dehydrogenase. Now this is a cytosolic version of this. So this is going to take dihydroxyacetone phosphate and it's going to convert it to glycerol 3-phosphate. Now this can go through the membrane, but then in the inner mitochondrial membrane, you have another isozyme of this enzyme right here, and this is GPD2. So this is glycerol 3-phosphate dehydrogenase, and that's going to take this molecule here, and it's going to reconvert it back to dihydroxyacetone phosphate, and it's going to give those electrons to a flavine, and it's going to make a reduced flavine. But this now is inside the mitochondria. So these reducing equivalents from glycolysis ended up getting over here via this shunt here that takes things, reducing equivalents from dihydroxyacetone phosphate to glycerol 3-phosphate back again, and then these reducing equivalents are given to the flavine here in the mitochondria. This is called the glycerol phosphate shuttle. Glycerol phosphate shuttle. Any questions about that before we move on? No questions, and we will resume. Why on earth does it do that? Okay, so here are some reactions catalyzed by these NAD and NADP linked dehydrogenases. This simply shows you where they're located, whether they're located in the mitochondrion or in the cytosol. Notice some come as two isozymes. The isocitrate dehydrogenase comes in a mitochondrial version that uses NAD. The cytosolic version uses NADP because it's used predominantly in anabolism. Malate dehydrogenase, both mitochondrial and cytosolic, uses NAD. So flavoproteins. Flavoproteins are one of the sources of electrons, but since FAD or FMN are tightly bound to their enzyme platforms, they don't directly participate in electron transport. Instead, they're going to pass their electrons from one source to another, or they may pass their electrons to NADH to make NAD to make NADH. This is precisely how pyruvate dehydrogenase works. It reduces flavine, and the flavine is used to reduce NAD to NADH. As we said here. So during electron transport, the electrons pass through electron carriers that are embedded in the membrane. These carriers are arranged in a sequential fashion, and they are structured such that they're in an almost solid state structure. Therefore, the transfer of electrons is extremely fast. Now, these carriers, some of them can carry one electron. Other carriers can carry two electrons. They're going to alternate between those who carry one and those who carry two. We'll see why in just a bit. Those carriers that carry but one electron very often will make use of iron. They will reduce iron from iron 3 to iron 2. From iron 3 to iron 2. They'll go from ferric to ferrous iron. Iron cannot carry hydrogen ions as well. It can just carry electrons. So those carriers that use iron are only going to carry an electron. Other types of carriers include ubiquinones, cytochromes, and iron sulfur proteins. Ubiquinones, cytochromes, and iron sulfur proteins. Let's look at these three carriers. Ubiquinones are also called coenzyme Q. Coenzyme Q. Coenzyme Q is used as a health food. If you go to health food stores, you'll see coenzyme Q on the shelf. When my sister did a good deal of thing, coenzyme Q was kind of all the rage. It was supposed to be cheater pills. I don't think it's recognized as cheater pills anymore, but this is a lipid-soluble molecule that has a long isoprene chain. That isoprene chain inserts it in the membrane, and then it has a head, and that head is a benzoquinone top. Because of its lipid solubility and small size, this can shuttle protons and electrons between complexes in the electron transport chain. This is a very heavily used carrier in electron transport. Ubiquinone can carry both protons and electrons. Many proteins in the electron transport chain will also have little pockets where ubiquinone can bind. So this is very useful. It's very heavily used. This will take your electrons where they need to go, one place to another also. You can carry singlet electrons and single hydrogen atoms as semiquinone radicals that, because of resonance, are somewhat more stable than other radicals. So here's a picture that shows you the ubiquinone. You see here the long isoprene tail. Here's the benzoquinone here. So this is the fully oxidized ubiquinone. You give it a single electron, single proton. You make a semiquinone radical. This is subject to resonance stability. Then you give another proton electron. Now you make an aromatic ring. This is ubiquinol. This is fully reduced. And it can unload those electrons back to a ubiquinone. It can do it sequentially though. And this can actually exist for a longer period of time than you think, the semiquinone radical. Very, very useful molecule when it comes to electron transport. Plants have a similar molecule, except it's slightly different structurally. It's called plastoquinone. Plastoquinone. Cytochromes. Cytochromes contain heme groups. These heme groups are tetraperols. They're made via a pathway that begins with succinyl coenzyme A. In the center is an iron atom, ion actually. The iron is the business end of this molecule. There are three types of cytochromes: cytochrome A, B, and C. Cytochrome A, B, and C, they're shown here. The A-type cytochrome has heme group to which a isoprene chain is attached. So this will be able to associate with a protein via hydrophobic interactions that often include the membrane. These hydrophobic interactions tend to be pretty strong, but this is not covalently linked to the enzyme. It's linked via hydrophobic interactions. That's an A-type cytochrome. B-type cytochromes, as you can see, have these two hydrophobic groups and then these two charge groups. These are associated with the protein via non-covalent interactions, the B-type cytochromes. By the way, A and B-type cytochromes are virtually always integral membrane proteins. C-type cytochromes are sometimes peromembrane proteins, but very often they can also be, they're peripheral membrane proteins, but they can be integral membrane proteins under certain circumstances. C-type cytochromes, as you can see, have the heme group covalently linked to the polypeptide chain. The standard reduction potential of the cytochrome is dependent upon the interaction with the protein side chains that hold it in place. So the reduction potential of cytochromes can vary pretty substantially. Here is the spectrum of a reduced and oxidized C-type cytochrome. And as you see, round about 420 or so, you have this difference in absorption in an oxidized and reduced C-type cytochrome. You also, with the reduced cytochrome, have this very strong absorption peak at about 560. Those are used essentially to determine if your C-type cytochrome is oxidized or reduced. All right, I said that already. What's the delta E of these? It depends on the polypeptide chain that holds them. So C's can vary. C-type cytochromes can be peripheral, they can be integral, it depends. A and B-type cytochrome are almost always integral membrane proteins.
Could I ask a question?
Oh, why not?
Well, I just want to make sure I got this right. For heme A, the isoprene chain is associated with a protein via hydrophobic interactions, and the protein.
Yeah, or it's stuck in the membrane,
okay, so
and the
either
heme, just the heme, interacts with the protein. You see both types.
is the protein, if it is associated with a protein, is the protein an integral membrane protein?
Yeah, virtually always.
Okay.
Virtually always. In fact, I really can't think of a case off the top of my head where an A-type cytochrome is not integral. Now, there might be one out there that I don't know about. It's entirely possible, but it's definitely in the minority. Okay, iron sulfur proteins come in one of three different forms. So typically, these will consist of iron molecules that are in a dative set of bonds with sulfur atoms that are often supplied by cysteine residues. And you can see these here. So you have kind of a simpler sort of structure where you just have 4 cysteines in a loop structure. Those coordinate to make the iron bind to the sulfurs provided by the cysteine. Or you can have something a bit more elaborate. Again, 4 cysteines, except you have kind of a, this isn't typically a beautiful planar structure, but it's a kind of a cube that somebody kicked in the teeth, or square that somebody kicked in the teeth. And then you have something a bit more complicated and frankly very beautiful, where you have these cubical sort of structures here. Then there's a special type of iron sulfur protein called a rescue iron sulfur protein. In this case, you have 2 histidine residues and 2 cysteines that work to coordinate the iron. When iron sulfur proteins participate in electron transport, they're only able to carry electrons, not hydrogen ions, just electrons. The reduction potential of iron sulfur proteins varies pretty wildly. It'll vary from 0.65 volts to plus 0.45 volts. Again, it depends upon the protein microenvironment of the iron within the cluster. So the standard reduction potential of these molecules also varies pretty substantially. How do you experimentally determine the order of the electron transport carriers in the electron transport chain? You use inhibitors. These inhibitors will bind to certain carriers and prevent them from shipping electrons to the next participant in the chain. So essentially, all the carriers downstream of that inhibited carrier are going to be reduced, and they'll stay reduced. All the carriers upstream of it will be oxidized, and they'll stay oxidized, and they can't be reduced. And by doing multiple rounds of these with different inhibitors, you can get an idea of which one comes first, which one comes second, which one comes third, so on and so forth.
Dr. Baranovich,
Yeah.
could you define the upstream and downstream for me, please?
Sure. Oh dear, that bled through. So this is an electron transport chain, and let's say you have a carrier here, you have a carrier here, you have a carrier here, a carrier here, and a carrier here. Let's call this 1, 2, 3, 4, and 5. So if you inhibit this carrier here, then the ones downstream will be reduced. All of these will be oxidized. If you inhibit this carrier here, all of these downstream will be reduced, and this one upstream will be oxidized. If you inhibit this carrier here, all of these downstream will be reduced, and these will be oxidized. So if you're moving the electrons this direction, upstream means towards the terminal electron carrier. Downstream would be towards the source of your electrons. That's downstream, that's upstream.
Okay, thank you.
Very good. So we use these carriers, and here are some of the carriers that have been used historically. Some of these are kind of famous. I don't know if any of you are Agatha Christie novel enthusiasts. Amytal, sodium amytal, is a poison that you see in her novels. So amytal, which is a barbiturate, for example, can prevent the transfer, essentially, of electrons from an iron sulfur center to ubiquinone. So can rotenone, which is a rat poison. There's a antibiotic called ptericidin A. You can see ptericidin A, that, like amytal, prevents transfer from the iron sulfur center to ubiquinone, and so on and so forth, and blah, 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 and what have you. And cyanide and carbon monoxide inhibit cytochrome oxidase. You get the point here. So, for example, there is a particular one of these. In fact, it's this one here. These three inhibitors here will prevent the reduction of all electron transport elements, and it blocks the further oxidation of NADH. That's going to suggest that NADH is the first element in the electron transport chain, since the initial oxidation of NADH occurs, but then is very quickly blocked. And so you can see essentially how you might do this. So you have tools to be able to determine which of these is reduced, which of these is oxidized. Use rotenone. These all stay oxidized. That tells you NADH. Well, that happens at any. Antimycin C, you get these three being reduced. All of these down here, they're oxidized. You use cyanide or you use carbon monoxide, and all of these will be reduced, but you don't get production of water from molecular oxygen, and so on and so forth. And by using combinations of these, you can order essentially the components in the electron transport chain. All right, so if you interpret these data essentially show you NADH is first, and then the reduction of water is last, and then antimycin somewhere in between. And then by using other tools, you can figure it out. Essentially, this is how the order of the electron transfer chain is figured out. So you take the mitochondrion, you then mix it, you break them open, you take the membrane fragments, use detergents to make various fractions of them, and you have acid to be able to reduce one to the other or something like that. And then you ask, if we put these together, which of these gets blocked by what, and so on and so forth. And yeah, that's how this was done. All right, so this sort of very rigorous biochemistry established that there are 4 complexes that compose the electron transport chain in mitochondria. And they're given names that only a biochemist could love. They're called complexes 1, 2, 3, and 4. And they're labeled with Roman numerals 1, 2, 3, and 4. Now, 2, as you can see, is oxidate dehydrogenase. So if your electron carrier is NADH, you're going to use complex 1, and then complex 1 will pass its electrons to complex 3, which will then pass its electrons to complex 4. If your carrier is reduced flavine is given to complex 2, which then passes its electrons to complex 3, which then passes its electrons to complex 4. So you don't go 1, 2, 3, 4, you go 1, 3, 4, or 2, 3, 4. And that's based on this type of very rigorous type of biochemistry that orders the components in the electron transport chain. Now, an electron transport chain has 3 main requirements. Here's the first requirement. They have to be embedded in a membrane. That membrane has to be impermeable to hydrogen ions. Must be proton impermeable. Second requirement. You must have the carriers in an order by which they are arranged by descending electronegativity. So that means the first items in the electron transport chain will have standard biochemical reducing potentials that are relatively negative. And then as they go down the chain, they will get progressively more positive in their standard biochemical reduction potentials until you get to the end, and that particular carrier will have a standard biochemical reduction potential that is quite positive. This way, the flow of electrons down the chain is thermodynamically spontaneous. Thermodynamically spontaneous. 3, here's the third requirement. You have to arrange the elements in the electron transport chain so that they alternate between carriers that carry just protons, carriers that carry just electrons, and carriers that carry both electrons and protons. You don't have this arrangement, then this is not going to work. And I'll show you why here in just a bit. So here is the path of the electrons through these complexes. Again, complex 1 gives us electrons to ubiquinone and reduces it to ubiquinol. Complex 2 gives its electrons to ubiquinone, reducing it to ubiquinol. Ubiquinol then takes electrons to complex 3. Cytochrome C then takes the electrons from complex 3, complex 4. Complex 4 is a cytochrome oxidase that will take the electrons and reduce molecular oxygen to water. This is a very good place to stop. So we'll stop here. We'll take a look at each complex and all its glory. These complexes are very complicated, but they're elegant in what they're able to do. Your quiz for Friday will be on Blackboard, and it'll be over the Krebs cycle. The chapter that covers the Krebs cycle, I believe, is chapter 16, if I'm not mistaken.
Will that include the glyoxylate cycle or not?
Yes, it will. It'll include the oxalate cycle, which is part of chapter 16. And we will pick up here on Friday. So I bid all of you a good afternoon, and I will see you all on Friday.
I have
Okay?
a quick question.
Yeah.
So I have a question on the test. It's number 3C. I'm just confused because I'm not very, I guess I haven't gone on the protein data bank website, but can you just show me how to get to the protein that you want us to look at?
Okay, what's the number I give you?
You say use the PDB in identifier 1DEP to retrieve data page for a portion of
1DEP.
Yes.
There it is.
And is that, because then it says to retrieve the data page for a portion of the beta and adrenergic receptor. Is that
Adrenergic.
Is that what we're looking for right here? The 1DEP?
This is part of the beta adrenergic receptor.
Okay.
See this here?
Yeah.
It's the third intracellular loop of the beta adrenal receptor. That's what that is.
Okay, so that right there is what we're looking for. And that's
Yep.
what the question's over.
That's what you're looking for.
Awesome. Thank you.
Okay. All right. See you, gang.